Welcome back to another video. This is the 9700 May-June 2018 paper 23. If you are new to this channel, you know what to do. Like, comment, subscribe and finally watch and learn. Without wasting further more time, let's get into it. Question 1. Figure 1.1 is a photomicrograph of root tip meristem. Different stages of the cell cycle are visible. Some cells are in the same stage of the cell cycle and some are in the same stage of mitosis. Part A. Cell D and cell E are in the same stage of the cell cycle. State the difference between the nucleus of cell D and cell E. Let's look at cell D and cell E. Well, if you look at cell D, you can see clearly that there are two nucleoli, whereas in cell E there is only one. Therefore, the difference that is visible and that is clearly shown in this given diagram is that the nucleus of cell D has two nucleoli, whereas cell E has only one nucleoli. And that earns you 1 mark. Part B. Name the stage of mitosis occurring in each of cells A, B and C. Let's look at A, B and C. Well, cells A and B are in the same stage of mitosis because the question does state that some cells are in the same stage of the cell cycle and some are in the same stage of mitosis. And what are the stages of mitosis? The stages of mitosis are prophase, metaphase, anaphase and telophase. And if you look at A and B, well, you can clearly see the chromosomes being visible and it does represent at the start. Therefore, A and B does have to represent the stage known as prophase, whereas A you can mention it as early prophase and B late prophase, because first the chromosomes start to be visible, which is shown by A, and then it does appear somewhat like cell B towards the end of prophase, hence known as late prophase. Therefore, A is early prophase, B is late prophase, and what is C? C shows the separation of the chromatids. And what does that stage represent? That stage is known as anaphase, because anaphase is where chromatid separation occurs. Therefore, C is anaphase. As you can see over here, A early prophase or prophase, cell B late prophase or just prophase, and cell C is anaphase. Part C. Cells F and G are newly formed cells. Cytokinesis has occurred within the formation of a cell plate. Describe the events that have occurred in the stage of mitosis immediately before cytokinesis. The stage of mitosis that is immediately before cytokinesis is known as telophase. And what happens in telophase? The spindle fibers are broken down, nuclear envelope reforms around the cell, as well as chromosomes uncoil and loosen. Basically, what happens in telophase is the opposite of prophase. Remember that carefully. Then it is easy to write down the events that occur in telophase. In prophase, the nuclear envelope breaks down, then spindle fibers start to form, chromosomes become visible by coiling, and the opposite of that is what happens in telophase. Hence, spindle fibers are broken down, nuclear envelope reforms, chromosomes uncoil and loosen. Writing 2 of these given points would earn you 2 marks. Question 2. Adipose tissue, which is composed of cells known as adipocytes, stores large quantities of triglycerides and functions as an energy storage tissue. Figure 2.1 is a photomicrograph of adipose tissue. Part A. Adipocytes can be very large in size compared to other body cells. This is due to a large lipid droplet within the cell. The largest adipocyte in figure 2.1 has a mean diameter of 35 micrometers. A person with good eyesight can see cells of 0.05 mm or greater diameter without a magnifying glass or any optical aid. State whether the person can see this adipocyte without any optical aid. Show your working to justify your answer. Obviously, if you do know how to work out the resolution as well as whether it is within the resolution limit, then you will be able to answer this question. The answer is, the person is unable to see the adipocyte as the largest, which is given in figure 2.1, has a mean diameter of 35 micrometers, whereas a person with good eyesight has 0.05 millimeter as its resolution. And in order to compare whether it is within the resolution limit or not, you have to convert it into a single unit. Then only you can compare and form a conclusion. Therefore, first let's convert 35 micrometers into millimeters, and you would get 0.035 millimeter, because 1 millimeter is equal to 1000 micrometers, and if you carry out the math, you will get 35 micrometers in millimeter as 0.035. Then 0.035 is less than 0.05 millimeter. In other words, the diameter of the adipocyte is smaller than the resolution size of the eye, hence it is not visible. Remember this carefully. If a given cell is smaller than the resolution size or less than the resolution limit, then the person will be unable to see it. A person can only see cells that have a diameter which is greater than the resolution limit. Since this given adipocyte has a diameter which is less than 0.05 millimeter, the person requires optical aid. Part B. Only some of the organelles within the adipocyte can be seen using a high quality light microscope set at the highest magnification. Organelles such as rough endoplasmic reticulum, smooth endoplasmic reticulum, and ribosomes are only visible using an electron microscope. Explain why these organelles are not visible using a light microscope. This right here has to do with the first chapter of your textbook, where light microscopes and electron microscopes are involved. And obviously, in a light microscope, the resolution is smaller. Remember that. Therefore, the ability to distinguish between materials in a light microscope is less. The organelles mentioned over here, such as rough endoplasmic reticulum, smooth endoplasmic reticulum, and ribosomes, have a size that is smaller than the resolution of the light microscope, hence not visible. Again, as we discussed in the previous question, those organelles or cells which are less than the resolution limit will not be visible, and those which are greater than the resolution size will be visible. And why are these organelles not visible using a light microscope? It is because the resolution is smaller. The ability to distinguish between materials in a light microscope is less. You have to link the resolution with the ability to distinguish between materials. If the resolution is greater, then the ability to distinguish between materials is high. If it is smaller, then it is the opposite. As well as, organelles are not visible because they have a size smaller than the resolution of the light microscope, and that will earn you 2 marks. Part C. Adipocytes synthesize triglyceride lipase, an enzyme that catalyzes the formation or breakdown of triglycerides as shown in figure 2.1. The balance between triglyceride formation and breakdown is controlled by hormones. Figure 2.2 is a summary of events occurring in an adipocyte when glycogen energy stores have been used up. Part 1. Name the type of bond broken by active ATGL to produce fatty acids and glycerol. Active ATGL is the enzyme which is known as triglyceride lipase. And active ATGL is involved in breaking down triglycerides to fatty acids and glycerol. And what are the bonds present in triglycerides? The bonds present in triglycerides are known as ester bonds. So what must be broken is definitely an ester bond. Therefore, always remember that it is the ester bond which holds the fatty acids and glycerol together. Hence the answer is ester bond. Part 2. Name and outline the process by which the fatty acids shown in figure 2.2 exit the cell. The fatty acids shown in figure 2.2 exit the cell by using a transport protein. As you can see over here, the dotted arrow marks over here pointing from fatty acids then through the cell surface membrane towards the tissue fluid represents the process known as facilitated diffusion. Why is that? Because transport proteins are involved and it doesn't seem to require any energy because there is no mention of energy being involved or ATP being involved. And remember, transport proteins being involved means only 2 things. It can be either active transport or facilitated diffusion. It cannot be active transport over here in this case because active transport uses energy, whereas the other, which is facilitated diffusion, doesn't. Hence the process shown over here is facilitated diffusion. In order to land 3 marks, you should write the name of the process, which is facilitated diffusion. And how do you outline the process? You should mention there is an involvement of a transport protein which doesn't require any energy, and that will give you 3 marks. Part 3. Figure 2.2 is an example of cell signaling within the body. With reference to Figure 2.2, outline the process of cell signaling. So if you look at Figure 2.2, you can see that there is a hormone being involved which binds with a sort of a receptor on the cell surface membrane. Then it causes some series of reactions such as second messenger molecule being released, then inactive enzyme turn into active enzyme, and so on until the final process is reached. Therefore, how do you explain this cell signaling process? Well, you should mention cell signaling definitely involves a signaling molecule that in this case is a hormone. Then it does involve a receptor, target cells, and hence finally a response. Therefore, how do you outline the process? Well, you should mention a specific hormone which acts as a signaling molecule binds to a complementary cell surface receptor. Specific hormone which acts as a signaling molecule will give you 1 mark. Binds to a complementary cell surface receptor, another mark. Binding triggers reactions within the cell such as a cascade of enzymes. That will give you your 3rd mark. And finally, hence it leads to a response by the adipocyte which is a target cell. And that gives you your full 4 marks because 4 points are mentioned. Again, how do you outline the process of cell signaling? You should definitely remember signaling molecule binding to a specific receptor which is complementary in shape. Binding will trigger the reactions within the cell such as a cascade of enzymes as shown in Figure 2.2. Then that leads to a response by the adipocyte which is a target cell. Part D. The fatty acids released from adipocytes are transported in blood plasma and are taken up by cells. Although most cell types can metabolize fatty acids to synthesize ATP in the presence of oxygen, red blood cells cannot do this. Suggests why red blood cells cannot metabolize fatty acids to synthesize ATP. In order to synthesize ATP, there should be mitochondria because mitochondria is what provides the energy to make ATP. However, red blood cells do not have any organelles such as mitochondria to synthesize ATP, so you could mention that, landing you 1 mark. Or you could also mention that there is no nucleus in red blood cells, therefore no mRNA produced. You could also mention the required enzymes are not present. So writing any of these given points will give you 1 mark. Question 3. Nerium oleander is a xerophytic plant. A photomicrograph of a section through the leaf of Nerium oleander is shown in Figure 3.1. Part A. The leaf shown in Figure 3.1 has a number of adaptations to reduce water loss by transpiration. 2 of these adaptations are a multi-layered epidermis, stomata only found in depressions known as stomatal crypts on the lower surface of the leaf. Explain how a multi-layered epidermis and stomatal crypts will help to reduce water loss in N. oleander. Multi-layered epidermis. Presence of multi-layered epidermis helps in reduced cuticle transpiration, that is, water vapor lost through the cuticle is reduced, as well as it leads to increased distances for water to diffuse, hence there would be lower rates of photosynthesis, and that is how it helps to reduce water loss. Presence of stomatal crypts, well, it traps a layer of air or creates an area of moist air, reduces the effect from winds and external air currents, therefore reduced exposure and surface area to the surroundings, hence less water is lost. Water potential gradient becomes less steep therefore, hence it helps to reduce water loss in N. oleander. Writing these points will earn you 3 marks. Again, multi-layered epidermis. By the mention of the word multi-layered, you get the idea that there is an increased distance for water to diffuse from. Hence there will be lower rates of photosynthesis because increased distances leads to lower rates of diffusion. Lower rates of diffusion means less water vapor escapes the plant. You could also say reduce cuticular transpiration, that is water vapor which is lost from the cuticle due to its increased distance. Presence of stomatal crypts is where there will be a layer of air trapped near the leaf. This reduces the effect from winds and external air currents, thus reduced exposure and surface area to the outside surroundings, hence less water is lost. Sucrose, amino acids, and other assimilates synthesized in palisade mesophyll cells of N. oleander pass to the vein, where they can be transported within specialized cells from the source to the sink. Part B. Name the cells specialized for the transport of assimilates in N. oleander. Always write in full form, that is, phloem sieve tube elements and not phloem only. And yes, phloem sieve tube elements are those cells that are specialized for the transport of assimilates, not xylem vessel elements. Xylem is only concerned with the movement of water and mineral ions. However, for the transport of assimilates or organic substances, phloem sieve tube elements is the only cell which is specialized for its transport. Therefore, always write in full form, that is, phloem sieve tube elements, not just phloem. Part C. Explain the difference between a source and a sink. Remember these key points. A source is where assimilates are synthesized or made. A sink is where assimilates are stored and areas where the assimilates are used for growth. Examples of sinks are roots for storage of starch or developing buds. Again, a source is where assimilates are synthesized. Some sources being a leaf of a plant, where photosynthesis is occurred and the plant makes its own food, hence the leaf being a source. A sink is where assimilates are stored and areas where the assimilates which are synthesized in the source are used for growth. And obviously examples of such sinks are the roots, since the roots are unable to photosynthesize or make its own assimilates, therefore it will store the assimilates made from the source. This therefore would give you 2 marks. One of the enzymes involved in the synthesis of sucrose in the cytoplasm of palisade mesophyll cells is known as Cy-FBPase. The gene coding for this enzyme is Cy-FBP. The importance of Cy-FBPase in plant growth can be investigated using plants with a mutation in gene Cy-FBP. These plants cannot synthesize Cy-FBPase. Part D1. State what is meant by a gene mutation. A gene mutation is nothing but a change in the nucleotide sequence, or you could also mention a change in the bases. Writing a change in the nucleotide sequence or a change in the bases would earn you 1 mark. Part 2. Suggest one way in which the mutation of Cy-FBP prevents the synthesis of Cy-FBPase. Any suggestion related to preventing protein synthesis, since Cy-FBPase is an enzyme and all enzymes are made up of proteins. You could mention that no mRNA is produced, hence if mRNA is not produced, then no translation would occur, which is a part of protein synthesis. Or otherwise you could also mention if mRNA is produced, it is unable to bind with the ribosome. Writing any of these points which is related to preventing protein synthesis is a way in which the mutation prevents the synthesis of Cy-FBPase, because Cy-FBPase is an enzyme and all enzymes are made up of proteins. Part E. Monoclonal antibody can be produced that is specific to Cy-FBPase. This antibody is used by investigators to check that the plants with the Cy-FBP mutation do not synthesize this enzyme. Part 1. In monoclonal antibody production, a small mammal is inoculated with Cy-FBPase and several weeks later, cells are removed from the spleen. Some of these cells are required for the production process. Describe the events occurring within the body of the small mammal that lead to the formation of the cells needed for monoclonal antibody production. In this question, you have to mention about the monoclonal antibody production process. So once the enzyme, which is Cy-FBPase, is introduced into the body, it is recognized as a non-self antigen by the body, hence stimulating an immune response. When an immune response occurs, T cells in the body recognize this enzyme as foreign and releases cytokines, because the role of T cells is to release cytokines which stimulate B cells. And what do B cells do? Well, they produce plasma cells, which in turn produce antibodies specific in nature to the enzyme. B cells form plasma cells by clonal selection and clonal expansion. These plasma cells are collected by the investigators to be used for monoclonal antibody production. Writing these points will earn you 4 marks. Again, how does the monoclonal antibody production work? There is an involvement of a foreign substance or a foreign antigen introduced into the body, which stimulates an immune response. What happens in that immune response? T cells release cytokines which stimulate B cells to produce plasma cells. Plasma cells secrete antibodies specific in nature to the enzyme, and this process is done by clonal selection and clonal expansion. Remember the words clonal selection and clonal expansion in monoclonal antibody production. Then once these plasma cells are made, which secrete antibodies, the plasma cells are the ones which are required by the investigators. And if you refer to the textbooks, it is these same plasma cells which are fused with cancer cells to form hybridoma cells. Therefore, it is the plasma cells which are collected by the investigators to be used for monoclonal antibody production. And thus, these are the events occurring within the body of a mammal that lead to the formation of the cells, which are the plasma cells, needed for monoclonal antibody production. Part 2. Anti-Cy/FBPase monoclonal antibody is added to extracts taken from the leaves of the plants with the Cy/FBP mutation. State the expected results following addition of the monoclonal antibody that would confirm the absence of Cy/FBPase in the leaf extracts. Since the purpose of monoclonal antibodies is to bind with the enzyme in its presence, in the absence it will be no binding of antibody to antigen. Because monoclonal antibodies, they are specific in nature and they only bind to a specific substance, and in this case it is Cy/FBPase. If Cy/FBPase is absent, then that means there will be no binding of the antibody to the antigen, and that leaves you with your 1 mark. Part F. Investigations have shown that plants with the Cy/FBP mutation grow to a much smaller height and have proportionately far less starch stored in their roots than normal plants. Suggests why plants with the Cy/FBP mutation will store less starch in their roots. If there is a Cy/FBP mutation, there will be less enzymes, which are the Cy/FBPase, present to synthesize sucrose. As earlier in the question, if you recall, they have mentioned its involvement in producing sucrose. Over here, see, one of the enzymes involved in the synthesis of sucrose in the cytoplasm of palisade mesophyll cells is known as Cy/FBPase, and that is the enzyme which is involved in the synthesis of sucrose. Therefore, if there is a Cy/FBP mutation, there will be less enzymes present to synthesize the sucrose, therefore less sucrose transported from the source to the sink. If less sucrose is made, less sucrose will be transported from where it is made, that is the source, to the sink. Less sucrose present to be converted to starch in the roots, hence less starch in roots. And that is why plants with the Cy/FBP mutation will store less starch in their roots. Question 4. Oxygen enters the bloodstream from the alveoli in the lungs and carbon dioxide leaves the bloodstream to enter the alveoli. Most of the oxygen is carried by hemoglobin in red blood cells to the body tissues. Part A. Outline how oxygen enters the bloodstream from an alveolus. The process by which oxygen enters is through diffusion, as you all know. Diffusion is from a region of higher concentration of oxygen to lower concentration. Diffusion is also a passive process, which means it requires no energy or ATP. Diffusion occurs through the alveolar wall or across the squamous epithelial cells that line the alveoli. Writing 3 of these given points would earn you 3 marks. Since the question is outline how oxygen enters, you have to talk about the process, that is either diffusion or active transport or osmosis in this question. So since it is diffusion, you have to explain how it occurs. Diffusion is from a region of high concentration to a lower concentration, that is down a concentration gradient, and diffusion is a passive process which requires no energy. Then you have to state where it occurs. It occurs through the alveolar wall. Figure 4.1 is an oxygen dissociation curve for adult hemoglobin. The curve shows the affinity of hemoglobin for oxygen at the range of partial pressures found in the body. The values for plotting the curve are obtained in the laboratory by bubbling oxygen at different partial pressures through a solution of hemoglobin at 37 degrees Celsius and pH 7.4. At a different temperature or pH, the measured values will change, resulting in a different oxygen dissociation curve. So given over here is the oxygen dissociation curve at 37 degrees Celsius and pH 7.4. Part B. Figure 4.1 shows that the percentage saturation of hemoglobin changes at different partial pressures of oxygen. Part 1. Use figure 4.1 to calculate the difference in percentage saturation of hemoglobin at the lower partial pressure of oxygen of 2.7 kilopascals compared to the higher partial pressure of 13 kilopascals. Show your working. We had to calculate the difference in the percentage saturation of hemoglobin at these given partial pressures. If you look at 13 kilopascals, I have marked over here what the percentage saturation of hemoglobin is, and you would get around 97%. And at 2.7 kilopascals, you would get the percentage saturation of hemoglobin at around 31%. So remember the values, 97% and 31%. Therefore, 97% at 13 kilopascals and 31% at 2.7 kilopascals. Therefore, the difference would be 66%. Hence, the answer is 66. Part 2. Explain the advantage of having a difference in percentage saturation of hemoglobin at lower and higher partial pressures of oxygen. First, you must mention where it is higher. So, the higher partial pressure is in the lungs, obviously. Since we are talking about the partial pressures of oxygen, it will be highest or higher in the lungs and lower in the respiring tissues. What are the advantages of it? The advantages are at higher partial pressures, the oxygen binds with hemoglobin and it is released at lower partial pressures, thus allowing oxygen to be delivered to areas where it is lower in concentration, such as in respiring tissues. And as I mentioned, therefore aids in the removal of oxygen from hemoglobin in areas where there is low partial pressure, such as in actively respiring cells. Another advantage is that if there is a difference, it will determine the affinity of hemoglobin towards oxygen. The affinity of hemoglobin for oxygen is high in higher partial pressures and low in lower partial pressures. Writing these key points will earn you 3 marks. In a person with sickle cell anemia, the ability of hemoglobin to transport oxygen and carbon dioxide is severely affected. The cause of this disease is a mutation in the gene coding for the beta-globin polypeptide of hemoglobin. Part C. Define the term disease. A disease is nothing but an illness of the body that leads to poor health, characterized by a set of signs and symptoms. Writing that one statement will earn you 2 marks, because 1 mark is for the illness of the body that leads to poor health. The second mark is for characterized by a set of signs and symptoms. You could also mention reduces the effectiveness of functions. However, it is better to remember this given definition over here. A disease is an illness of the body that leads to poor health, characterized by a set of signs and symptoms. Part D. Outline the differences between the normal and sickle cell alleles of the gene coding for the beta-globin polypeptide and explain how these differences lead to a change in the hemoglobin molecule formed. In sickle cell allele, there is a base substitution which led to a different amino acid binding to the polypeptide chain which is being formed. So, due to the base substitution, there will be an altered mRNA codon. And since the question asks and explain how these differences lead to a change in the hemoglobin molecule formed, we have to state these given points. Base substitution results in mRNA codon being altered. Overall, there will be a change in the tertiary structure comparing the normal and the sickle cell alleles. In the end, the hemoglobin formed is less able to bind with oxygen. You could also mention which base is substituted. Thymine replaces adenine. This brings about the amino acid change from glutamic acid to valine in sickle cell allele. Remember these key points. The base substitution is where thymine replaces the adenine. This causes the amino acid change from glutamic acid to valine in sickle cell allele. Writing these points will earn you 5 marks. Therefore, we have tackled this question by breaking it into 2 parts. Outline the differences is where there is a base substitution. Explain why this base substitution leads to a change in the hemoglobin. Well, a base substitution results in an mRNA codon being altered. Then it affects the resulting amino acid binding to the polypeptide chain. So the amino acid is changed from glutamic acid to valine in sickle cell allele. This is due to the base substituted, which is thymine replaces adenine. Then in the end, there is a change in the tertiary structure of the polypeptide formed when you're comparing between the normal and the sickle cell alleles. In the end, hemoglobin formed is less able to bind with oxygen. Question 5. The fig tree and the papaya tree produce a milky looking fluid known as latex. The latex is released when plant tissue is wounded, and it is thought to act as a defense against attack by herbivorous insects or parasitic worms. Latex is a complex mixture of substances, and the exact composition of the mixture depends on the plant species. A group of enzymes that hydrolyze proteins known as cysteine proteases are commonly found in latex. Ficin found in F. cassia and papain found in C. papaya are both cysteine protease enzymes. These enzymes have been extracted and purified for use commercially. Part A. An investigation was carried out to compare the effect of temperature on the activity of ficin and papain. The results are shown in figure 5.1. Part 1. With reference to figure 5.1, describe the differences between the activity of papain compared to the activity of ficin between 20 degrees Celsius and 80 degrees Celsius. So obviously, if you go to this given figure over here, you can compare between the two their percentages of maximum activity as well as their temperature range. So the enzyme papain has a higher optimum temperature than ficin. As you can see over here, papain it is around 60 degrees Celsius, and for ficin it is around 45 degrees Celsius. In papain, the optimum temperature is 60 with 100% activity, whereas in ficin it is 45 degrees Celsius at 100% activity. Since the question is a describe question, you have to mention values while also noting the differences between the two. The enzyme ficin works better at lower temperatures or lower temperature ranges, whereas the enzyme papain works at higher temperature levels. The enzyme ficin works better at lower temperatures than papain, at steeper activity levels than papain at temperatures below 55 degrees Celsius. Yes, as you can see over here, papain works at higher temperature ranges, whereas ficin works at lower temperature ranges, if you look at the steepness of the curves. Part 2. Ficin and papain have been shown to be effective in the digestion of parasitic nematodes. With reference to figure 5.1, explain which enzyme you would select to use in an oral medication for the treatment of human intestinal parasitic nematodes. Okay, if we refer to figure 5.1, the enzyme which is best would be enzyme ficin, because enzyme ficin works better at lower temperature ranges and at the normal body temperature, that is around 37 degrees Celsius. It is enzyme ficin which works best. Therefore, ficin, because it has a higher activity at the normal body temperature of humans at around 37 degrees Celsius. Right in that given point over here would earn you 1 mark. Part B. One commercial use of the enzyme ficin is the production of Fab fragments, antigen binding regions of mouse IgG antibodies for use in immunological studies. The process uses immobilized ficin to cleave the antibodies in the hinged region. Suggest one practical advantage of using immobilized ficin for this process rather than ficin free in solution. The key part of this question is suggest advantages of using immobilized ficin, and we have to write over here the advantages of using immobilized enzymes which are given in your textbook. So you could say the enzyme can be reused if it is immobilized, hence lower costs involved, and the enzyme does not mix with the products, therefore no contamination. In such questions, mention the benefits of using immobilized enzymes, such as longer shelf life, product is not contaminated with enzyme. Writing any of these given advantages would earn you 1 mark. Part C. Streptococcus pyogenes is a bacterium that can cause a range of diseases in humans. S. pyogene synthesizes streptopane, a cysteine protease that hydrolyzes structural proteins in human connective tissue. Part 1. Streptopane is secreted to the outside of the cell. State the term given to the enzyme that is produced by a cell and is then secreted to the outside where it has its action. Such enzymes are known as extracellular enzymes. Therefore, an enzyme that is produced by a cell but it works outside of the cell by being secreted to the outside, then it is known as an extracellular enzyme. Part 2. Suggest one example of a structural protein in connective tissue that can be hydrolyzed by streptopane. Examples of structural proteins are collagen or elastin. Structural protein mentioned in our textbook is collagen, so remember that carefully. It is safe to write collagen over here, earning you that 1 mark. Question 6. Part A. Figure 6.1 is a list of infectious diseases. Each of the statements A to D describes a feature that applies to one or more of these diseases. So the figure given over here has cholera, HIV, AIDS, malaria, measles, smallpox, tuberculosis, and I have marked towards the side what is the causative organism of this disease. So let's look at the question. For each of the statements A, B, C, and D, name all the diseases in figure 6.1 that match the feature described. A. The causative organism of the disease is a virus. Obviously, if the causative organism is a virus, then you should know that HIV/AIDS is definitely caused by a virus. Measles is caused by a virus known as morbillivirus. Smallpox is caused by a virus known as variola virus. Therefore, you have to mention all the diseases. Hence, you have to write HIV/AIDS, measles, as well as smallpox. Part B. The causative organism of the disease is a prokaryote. Remember, all bacteria are prokaryotes. Therefore, you have to mention diseases which is caused by a bacterium. Hence being cholera caused by a bacterium known as Vibrio cholerae, and tuberculosis caused by a bacterium known as Mycobacterium tuberculosis or Mycobacterium bovis. Hence the answer is cholera as well as tuberculosis. Part C. The disease is transmitted by a fecal oral route. For example, sewage containing the pathogen contaminates drinking water. And this obviously is cholera. And this is concerned with the chapter known as infectious diseases. Therefore, if the disease is transmitted by a fecal oral route, the disease which should immediately pop up into your mind should be cholera. Part D. The causative organism of the disease spends part of its life cycle inside an insect which acts as a vector of the disease. And as soon as you see the word vector, what should come into your mind is mosquitoes. And mosquitoes is linked with malaria. Therefore, the disease is malaria. And this would earn you 4 marks. Part B. Although many infectious diseases are caused by prokaryotic organisms, there are some that are caused by eukaryotic organisms. Complete table 6.1 to show some differences between a prokaryotic cell and a eukaryotic cell. In this table, we have to compare between prokaryotic cell and eukaryotic cell, and some key things which should pop up into your mind immediately is that prokaryotic cells have no nucleus, whereas eukaryotic cells, they do have a nucleus, and that nucleus is surrounded by a double membrane known as a nuclear envelope. Remember, if there is a double membrane surrounding a nucleus, the double membrane is known as an envelope, and since it is a nucleus, it is a nuclear envelope. Therefore, the answer being true nucleus, genetic material enclosed by a double membrane known as a nuclear envelope. Then let's look at the next row. In eukaryotic cell, they have linear DNA, whereas in prokaryotic cell, it is circular DNA. Remember that key point, circular DNA in prokaryotic cells, such as in bacteria, and linear DNA in eukaryotic cell, such as in animals. Then the differences between ribosomes is that in eukaryotic cell, they have both 70S as well as 80S ribosomes. 80S is present in almost most of the cells, 70S being in organelles such as the chloroplast if it is a plant cell, or mitochondria if it is an animal cell. Whereas in prokaryotic cells, it is only 70S ribosomes which are present. Therefore, 70S ribosomes only in prokaryotic cell, 70S and 80S ribosomes in eukaryotic cell. Next row, no double membrane bound organelles in prokaryotic cell, whereas in an eukaryotic cell there is double membrane bound organelles such as mitochondria or chloroplasts. Then the next row is regarding the cell wall. In eukaryotic cell, if a cell wall is present, it is generally containing cellulose or chitin, whereas the cell wall of a prokaryotic cell contains murine or peptidoglycan. Writing these given points over here would earn you your full 2 marks. And that is the end of this paper. Hope you found this video useful. If you did, don't forget to hit that like button and share with your friends. In that way, we can keep this squad growing. Thank you for watching and stay tuned for more.